Hello, and welcome to QIAGEN's webinar on optimized sample processing and digital PCR-based workflow for wastewater monitoring of SARS-CoV-2. My name is Dominic O'Neill. I'm the Director for Microbiome Product Development at QIAGEN. I will be discussing the sample processing, and my colleague, Colin Donohoe, will talk about the digital PCR-based workflow. Before we start, a quick legal disclaimer. Any products that we speak about here today are intended for molecular biology applications and not intended for the diagnosis, prevention, or treatment of disease. So on our agenda for today is an introduction on the importance of wastewater surveillance for SARS-CoV-2, and then we'll go into the wastewater testing workflow that we have been developing at QIAGEN, starting with the importance of inhibitor removal technology, discussing some of the concentration methods that we've looked at, and ending with the full sample preparation workflow. And then Colin will provide some digital PCR background, as well as data on digital PCR quantification of SARS-CoV-2 in wastewater. And at the end, we have time for questions. So there's been tremendous interest in wastewater testing for SARS-CoV-2 surveillance over the past year, and it's because the benefits of this technology are potentially immense. Cost has been estimated that it could save up to about $1 billion in monitoring programs by testing on a population basis rather than individuals. The sensitivity can be huge. You can find infected individuals in populations of 100 to up to 2 million people to potentially find outbreaks before they spread. And you can increase your coverage dramatically by looking at billions of people potentially monitoring through their sewage treatment plants rather than trying to do individual testing. So detection of viruses in wastewater has been known for quite some time, in particular for poliovirus, for example, and those techniques are adaptable very easily to SARS-CoV-2 detection. It's known that SARS-CoV-2 passes through the intestinal tract and is shed into the wastewater, and therefore testing wastewater with the associated fecal matter will pick up SARS-CoV-2 viral RNA if it's present from infected individuals in the catchment. And so for that, the wastewater treatment plant is going to be the natural place to do this testing, as it will gather in sample from an entire catchment area, giving you a broad cross-section of the population, and then looking directly at the influent sample coming in before it goes to any treatment that could potentially decrease the signal, aside from potentially looking at the sludge that is settled out, is going to probably provide the best sensitivity. And the sample needs to be taken, processed in some way because it's normally quite dilute, so it needs to be concentrated. The nucleic acids need to be extracted through different methods, and that's shown here on the right, different concentration methods, with whether you're working with the raw influent or whether you're working with settled sludge. And then for detection, there's a variety of methods, but for SARS-CoV-2, PCR-based methods and digital PCR-based methods are going to be the most common method used. And as mentioned in the slide earlier, the importance of SARS-CoV-2 wastewater-based epidemiology is to detect small amounts of SARS-CoV-2 in complex samples, perform surveillance for public health decisions, which will let you understand the extent of the community spread, provide early detection, which you can then potentially use to prevent community outbreaks, predict new infections and quarantine people as the outbreaks begin, and also very interestingly, it's potentially used to monitor variant spread. The variant should also be detectable in the wastewater, and with sequencing or digital PCR multiplexing, various variants can be detected. So for each step along the workflow for SARS-CoV-2 wastewater-based epidemiology, QIAGEN has solutions for dealing with collected samples, for disrupting the samples, for extracting the samples, for doing detection, and for doing data analysis. For example, using any of our manual kits or the automation options here for doing the primary sample extraction, followed by the Kai Acuity digital PCR instrument, and the Kai Acuity software suite for doing the data analysis provides a complete workflow. Now, I will talk in detail about the sample preparation portion of the workflow, so about collecting samples, about sample disruption, and sample extraction. So before I go into the data that I have to show, Just here a quick excerpt from a paper from September of 2020 from Dr. Warish Ahmed on surveillance of SARS-CoV-2 RNA in wastewater, talking about the methods of optimization. And here their conclusion is also that RNA extraction kits that are streamlined and that are equipped with PCR inhibitor removal technologies are going to be the most useful to reduce the chance of contamination. Wastewater samples are dirty. They have a huge amount of inhibitors present and are very, very difficult to get clean RNA and DNA out, particularly when you don't know what the status of the viral RNA is and how degraded it might be. And so that, of course, is very important. The inhibitors that are being talked about here are small chemicals that are released at the same time that you break open cells or when you disrupt your biological sample. These could be, for example, humic fulvic acids coming from soil, polysaccharides, polyphenolics in plants, bile, bilirubin, heme, and stool, for example, as well as others. And these are potent inhibitors of downstream PCR assays. On the right, you can see a PCR curve of a same sample processed with and without QIAGEN's inhibitor removal technology. And without the inhibitor removal technologies, those are the curves labeled as plus inhibitors, you can see a dramatic shift in CT to the right, where the samples are coming up much later. And this is due to these inhibitor samples. And this clearly is going to decrease your sensitivity when doing a PCR. You won't detect positive samples if you do not remove your inhibitors ahead of time. And so we looked at this. We looked to see how critical it is for wastewater samples to use IRT. So IRT is QIAGEN's proprietary inhibitor removal technology. It is present in any kit that has power in its name. So the All-Prep Power Viral, the RNeasy Power Water, the RNeasy Power Microbiome, for example, those all contain this IRT. Now the QIAGEN Viral RNA Mini Kit is standardly used for SARS-CoV-2 detection or extraction and analysis in swab samples, for example, but it's not designed to deal with fecal material or sewage samples. And here, when we compare it, we see that as well. Using different concentration methods, whether we spun down and processed a pellet or did ultra filtration or did electronegative filtration on a mixed cellulose ester membrane, we see a significant shift in CTs when we prep those materials with the QIAGEN Viral RNA Mini Kit, and we see much better consistent detection using either the All-Prep Power Viral or the RNeasy Power Water Kit. And this is an indication that there are potent inhibitors present in the sample that are removed through the use of IRT. So we strongly recommend any IRT kit when dealing with wastewater samples. So I mentioned on the earlier slide that any IRT kit will be recommended. So that's true for any of the RNeasy IRT kits. And we recommend multiple kits here because they use a very consistent chemistry. So the RNeasy Power Microbiome, the RNeasy Power Water, and the All-Prep Power Viral DNA RNA are all suitable for viral detection in wastewater. We have tested all of them on actual wastewater samples successfully. And the reason that they are interchangeable, as you can see in this chart here, they use similar lysis buffers, similar IRT, similar binding and washing. The differences are in the bead beating, which we have found not to be necessary for SARS-CoV-2 detection. The viral RNA is not protected in a way that requires bead beating to recover it. And DNA's treatment, since you're doing a specific RT-PCR at the end, is generally also not required. The DNA in this case will not interfere, and so we would generally leave that out. And in that case, these kits are basically the same and can be used interchangeably, with one caveat that I'll talk about later. So looking at the virus concentration methods, I talked in the previous slide that we had looked at pellets, we had looked at mixed cellulose ester membranes, and we had also looked at ultrafiltration. Those are all methods that are described here and as standard methods of doing concentration of SARS-CoV-2 in wastewater. We were interested to see whether there was one method that we could recommend over any other. So here you can see that we attempted to extract SARS-CoV-2 from samples concentrated over mixed cellulose membranes, ultrafiltration retentates, or through spinning down and processing the pellets. And we get similar detection from this wastewater sample in any three of the methods. So Any concentration method is theoretically compatible with our IRT kits and can be recommended. However, not all wastewater samples are equally amenable to all concentration methods. So what we also found here, looking at different wastewater treatment plants, is that different materials are going to be differently suited to the concentration method. So in this experiment, we looked at two different wastewater treatment plants. One of the plants was more of a municipal plant. It was collecting sewage that was flowing through the sewage system, had more solids, had more human material present. It created a very strong pellet. And so when we spun it down, we got a lot of material that we could process as a pellet directly into our kit. When we tried to process the material through the ultrafiltration membranes or through the mixed cellulose esters, those methods clogged and were not able to process more than 10-15 mLs compared to extracting out of 50 mLs when we were doing the pellet system. And as you can see, we got much better detection for that municipal plant when dealing with the pellet. The other plant that we were dealing with was a more general plant. It included water flowing over overland streams, had more environmental runoff, and that provided better results with the filtered material slightly. It had signal in the pellet as well, much more comparable between all three methods, and was also easily filterable through all of the methods. So the particular sample used will affect which concentration method is going to work best in your laboratory. The important note is that all of our kits will work well for all concentration methods. So our recommendations for doing RNA extraction from wastewater is to filter your sample. If you filter your sample, you should use the RNeasy Power Water Kit because that provides 5 ml tubes to do processing of the larger filter. If you're going to concentrate the sample through pelleting or through ultrafiltration, then the All-Prep Power Viral DNA RNA or the RNeasy Power Microbiome Kit work well. In our experience of testing different wastewater samples, bead beating is not necessary, also not harmful. This may vary depending on your particular sample, and particularly when doing filters, bead beating may help to scrape material off of those filters. And so we don't recommend that people do not do bead beating, but we do not find that it's necessary. Likewise, DNase digestion is not necessary when using these kits. There is a standard protocol for the RNeasy Power Microbiome Kit and a custom protocol for the All-Prep Power Viral Kit on the Kai Acuity. The custom protocol can be requested through our apps lab, and we are working on standardizing and releasing the All-Prep Power Viral also as a standard Kai Acuity process. And that brings me to the end of my slides. And here I'd like to just leave off with another excerpt from a paper by Dr. Ahmed, concluding that extraction of lysate using the RNeasy Power Microbiome and Kai Acuity Connect platform to obtain high quality RNA is highly suitable for wastewater analysis. Thank you for your attention, and I'll hand it over to Colin for the digital PCR portion. Thank you.
Thanks, Dominic. Now I'm going to tell you guys how you can use the Kai Acuity digital PCR system to detect SARS-CoV-2 in your wastewater samples. I'd just like to reiterate a few things that Dominic has already said about SARS-CoV-2 and wastewater testing before going any further. The real advantage of using wastewater samples to monitor SARS-CoV-2 is that within a single sample of wastewater, hundreds, thousands, and even millions of people are represented. And this allows researchers to get a much better snapshot of the community spread of SARS-CoV-2 when compared to looking at individual clinical samples. And this data from wastewater can be used as an early warning sign for outbreaks and allows researchers to get a snapshot of SARS-CoV-2 variant spread within a single sample. And they can relate the quantities of each variant to each other very easily. Now, digital PCR is particularly well suited to this task because unlike qPCR, digital PCR is absolute quantification where no standard curve is required, allowing data collected between various sites to be compared with great certainty and precision owing to the fact that the data is absolute in its quantification. Additionally, because digital PCR quantitates a little bit differently from qPCR, the chemistry is a little bit more robust to the wastewater samples, which are known to contain PCR inhibitors. So why is the Kai Acuity Nanoplate digital PCR system ideal for wastewater testing? First of all, the time to result with the nanoplate is only 2 hours, which is much shorter than the 5 hours it takes using the conventional digital droplet PCR system from Bio-Rad. Second, the Kai Acuity Nanoplate system is capable of high multiplexing, up to 5-plex, meaning that users can detect multiple SARS-CoV-2 variants within a single reaction well. Lastly, the Kai Acuity Nanoplate system is fully integrated in its workflow and offers users the flexibility of throughput and resolution. The plates are very similar to qPCR plates in their shape and well layout, and the 24 and 96 well plate options allow users the flexibility of throughput and resolution all at once. I'd like to take a brief moment to point out the features of the Kai Acuity Nanoplates and how they differ from the digital droplet PCR workflow. In the Kai Acuity Nanoplate, you can see your results literally in the raw image of each and every well on your nanoplate. Second of all, sealed nanoplates prevent contamination between wells. Once your reaction mix is in a well, it's in a closed environment and it stays there. The solid partitions, compared to the droplets, have a very reproducible size and therefore are particularly well suited for the Poisson calculations that underlie digital PCR, which rely on a distinct and discrete partition volume. Because we can image an entire well at once and not via the flow cytometry style of digital droplet PCR, the readout is fast, and because our plates are the standardized format of all PCR plates, the workflow is very simple, and anyone who's familiar with qPCR will have no problem jumping into the Kai Acuity Nanoplate world. Of course, in order to use the Kai Acuity Nanoplates, you need Kai Acuity Instruments. And here I'd like to talk about the 4 instruments that we have available. We have a single plate instrument called the Kai Acuity 1, which comes in 2 configurations. There's the 2-channel instrument or the 5-channel instrument. Next to that, we have 2 higher throughput instruments called the Kai Acuity 4 and the Kai Acuity 8, which can process 4 or 8 plates respectively in parallel. And both of those instruments come only in one configuration with respect to multiplexing, and that's the 5-channel system. On this slide, we have the consumables from QIAGEN, which you can use to detect SARS-CoV-2 in wastewater. As mentioned before, QIAGEN offers nanoplates in a variety of formats. 26K nanoplates contain 26,000 partitions per well, allowing users to load up to 28 microliters of sample per 40 microliter reaction. Alternatively, users can opt for the lower resolution 8.5K nanoplate, which comes in 2 formats, 24 well and 96 well, where 8,500 partitions per well are found, with reaction volumes of 12 microliters, where users can use up to 7 microliters of sample per reaction. We have a Kai Acuity one-step viral RT-PCR kit developed special for SARS-CoV-2 detection. It comes in one size, enough for 520 reactions in 26K nanoplates, and the kit contains the 4X master mix, 100X multiplex reverse transcription mix, and RNase-free water. It's not available on web shop ordering, but if you're interested, contact your QIAGEN sales specialists to order it. For the one-step viral RT-PCR kit, we came up with distinct single and multi-plate workflows. For users interested in only processing a single plate at a time, we came up with a single plate workflow which works on all Kai Acuity instruments. As is part of all Kai Acuity workflows, users assemble their reaction mixes in so-called pre-plates. And after they've set up their reactions in pre-plates, they transfer the reaction mix to a nanoplate, seal the nanoplate, and then put the nanoplate into the Kai Acuity instrument, where there, RT incubation and 2-step Kai Acuity PCR cycling take place without any additional user input. Now, for users interested in processing multiple plates in parallel using the Kai Acuity 4 and 8 instruments, we came up with a dedicated multi-plate workflow, owing to the fact that reverse transcriptases are not meant to be sitting around for the length of time that it would require if one were to load a full Kai Acuity 8-plate or 4-plate instrument and wait up to hours until the reverse transcription step in the cycling program took place. This is particularly important for the generation of nonspecific signal or spurious side products which you don't want happening at room temperature. Therefore, we came up with the idea that users would assemble their reaction mixes in pre-plates, which are anyways 96 well PCR plates standard, and they would perform an offline reverse transcription step in the pre-plate. After the reverse transcription took place, the user would inactivate the reverse transcriptase enzyme using a 10-minute inactivation step, then transfer their reaction mix to the nanoplate, and then perform the two-step Kai Acuity PCR cycling in the Kai Acuity instrument. Now we'd like to show you guys some application data where we quantified the abundance of SARS-CoV-2 using RNA extracted from German wastewater using the methods that Dominic described previously in the PowerPoint. So here below we have 9 distinct biological samples which were taken from the same pool of collected wastewater and were then extracted using MCE membrane or the concentration of retentat through Amicon filtration devices or centrifugation of wastewater to form pellets which were then extracted. We assayed the RNA using the N1 and N2 assays developed for SARS-CoV-2 by the CDC. We used 5 microliters of RNA in 40 microliter 26K nanoplate reactions, and the original eluate was derived from 50 milliliters of wastewater. As you can see, consistent with what Dominic saw, the pellet and Amicon retentat samples quantified SARS-CoV-2 better than the MCE membrane. We'd also like to show you guys some application data where we tried out some troubleshooting of techniques when dealing with wastewater samples because they can be tricky at times. And so we played with 3 variables. First, we increased the annealing extension time from 30 seconds at 60 degrees to 90 seconds at 60 degrees in our 2-step PCR system. Second, we investigated whether using less eluate per digital PCR reaction improved the quantification overall. Lastly, we tried adding a well-known PCR inhibitor magic pill, BSA, to see if that would also help. And as you can see, using the N1 assay, using less template, increasing the extension time from 30 seconds to 90 seconds, and adding BSA to the system all helped in increasing the detection of SARS-CoV-2 in our wastewater samples. So I would just like to recommend that if anybody's having difficulty with their wastewater samples, try playing with those 3 variables. Last but not least, we've also gathered from our experience in quantifying SARS-CoV-2 in wastewater that users coming from the qPCR world to the digital PCR world are sometimes surprised that their quantification expectations aren't matched when they come to digital PCR. And we found out that many times it's because concentration expectations determined with qPCR are determined using standards which are inaccurately quantified. Here we have 4 single-stranded RNA standards which were placed into digital PCR reactions at expected concentration of 1,000 copies per microliter. And as you can see, all 4 standards came below the expected concentration, and most importantly, among the standards, there were different concentrations, indicating that unique features to each standard were off. Each concentration for each standard was off, sometimes by a factor of 3, which would be quite significant for important quantification experiments. So here is QIAGEN's solution for SARS-CoV-2 detection in wastewater using extraction methods which we developed specially for this task, and the Kai Acuity instrument and the Kai Acuity one-step viral RT-PCR kit that I just talked about. So thanks for your attention, and we'll be taking your questions right now.